Welcome to part 2 of a series of 3 webcasts on isotopic analyses. This presentation covers isotopic water analyses by wavelength-scanned cavity ring-down spectroscopy, fast, easy and high-precision delta-18O and delta-D measurements from Picaro, producers of the world's highest performance and easiest to use analyzers. Isotopic analysis of water provides excellent information for scientists studying the climate, looking at historical changes in ice cores, and mapping water distribution and movement in aquifers and other sources. In addition, the isotopes of water can also provide information on movement of species, such as bird migration. In addition, if we consider the body as a water cycle system, delta-18O and delta-D values provide valuable information on metabolic activity. To understand these applications a little further, we will briefly review the water cycle and how isotopic enrichment processes occur naturally. Water in the oceans is made up of several isotopologues, among which are H218O, H216O, HD16O and other permutations. During the evaporation processes that form clouds, the lighter isotopes are preferred, which leads to the first stage of isotopic enrichment. As the clouds travel and move over continental land masses, condensation occurs, with the heavy isotopes being preferentially precipitated. That leads to clouds that are further enriched in the lighter isotopes or depleted in the heavy isotopes. Additional precipitation events repeat this process. So, in general, areas further from the oceans have water that is more depleted in the heavy isotopes. This leads to an abundance of potential applications. For example, there are mixing processes in above-ground and underground waterways that provide changing isotopic distribution. Animals and plants that feed and grow in a specific location may show an isotopic contribution definitive to that region. And the evaporation processes that form clouds have a temperature dependence that means that historical records of isotope values in ice cores can help us understand previous global warming and cooling trends. To emphasize the previous discussion, here is data acquired from 15 injections each of 5 groundwaters. The groundwaters range from Florida, Boulder, West Antarctica, Greenland, and Vostok, and show increasing depletion in both oxygen-18 and deuterium. If we look at an expansion of one of the samples, we see several important characteristics. First, the Picaro systems show minimal memory in both oxygen and hydrogen isotopic measurements. We attribute this to the design of our vaporizer unit, which will be discussed in more detail in an upcoming webcast. Second, the precision for both isotopes is equal to or better than typical IRMS data, 0.38 per mil for delta D and 0.03 per mil for delta-18O. Third, the memory and precision are as good at the end of the run as they were at the beginning, a span of approximately 12 hours without calibration. This performance is excellent news for users who would like to deploy this system in more remote laboratories or field stations. To take a further look at long-term performance, here is a suite of data collected over 3 days, again without calibration. The measured results from continuous injections of the same sample provided typical precision for delta-18O of 0.074 per mil and delta-D of 0.26 per mil. Data like this in turn provides us at Picaro with sufficient confidence to guarantee our instrument drift performance. Other laser-based products have anecdotally suffered from poor temperature stability, leading to a need for higher numbers of calibration standards. This in turn reduces throughput significantly. Picaro's small cavity design and excellent temperature control provide an analyzer with unsurpassed temperature stability. This chart again shows a series of continuous injections of the same sample made while the analyzer was running in a temperature-controlled chamber. The temperature was ramped from 33 degrees C to 41 degrees C, yet the data shows minimal change in delta 18O or delta D isotopic values. Again, this performance provides data credibility for samples run in remote labs or even ships or other forms of transportation, traditionally sites considered as challenging environments for IRMS systems or other laser-based spectrometers. Samples containing salts, such as those from oceanographic studies, inland lakes, or boreholes, are also suitable for this technology. Here's a split chart from a series of over 1,100 injections of a 4% by weight salt solution typical of seawater. On the left are delta 18O and delta D values for the first 10 injections. On the right are the delta 18O and delta D values from injections numbered 1,144 through 1,152. Two pieces of information stand out. First, there is no change in the values from the beginning of the run to the end. And second, the precision also remains incredibly stable for both isotopes, and well within our guaranteed performance for non-salty samples. We have also run higher salt concentration samples from inland lakes, containing from 160 grams per litre to upwards of 200 grams per litre of salt. The precision of the data is again well within our performance range, but more importantly, there is excellent correlation with IRMS data from the same samples. We have also run food samples which show interesting variations that may be related to both regional growing conditions and metabolic processes. First, let's look at two apple types. We bought Pink Lady and Granny Smith apples from a local supermarket. We cut a portion from each apple and squeezed about 150 microliters of juice from each into separate vials. We then distilled to dryness, capturing the water. A series of injections of each sample provided excellent precision. In this chart, the points in the same color represent multiple injections of the same sample, and precision is better than 0.08 per mil for delta 18O and 0.2 per mil for delta D. Additionally, the two apple types are clearly separated in this two-dimensional isotopic landscape, indicating that the apple types are differentiable by either regional or metabolic or both processes. Taking this application further, we performed a similar method on juices squeezed from two groups of oranges. The first set was purchased in California and the second set from Florida. Again, the precision was well within our typical performance, and also these two sets of oranges grouped convincingly. This time there was little change in oxygen 18 values, but there was a significant difference in delta D. We consider these results to be an excellent indicator of future applications, especially considering the incredibly simple method we employed. Our final dataset in this suite shows an isotopic urine analysis currently in development. This experiment requires dosing patients with doubly-labeled water, that is, water enriched in oxygen 18 and deuterium. The series consists of a set of baseline samples which are measured prior to dosing, followed by injections immediately after dosing which show very high isotopic values. Subsequent injections follow a time course and show a tracking back down towards the baseline numbers. One of the factors important in these labeling applications is the linear dynamic range of the instrument. There are many potential preclinical applications for this type of analysis. So, in summary, we have shown data that confirms the Picaro isotopic water analyzer is the no-compromise technique of the future. Customers have shown that our systems have performance better than IRMS, the total operating costs are significantly lower than other instruments, and that the instruments are easy to install and are transportable from one location to another. The remarkably low drift enables minimal calibration and highest throughput. Overall, these instruments are easy to operate even for demanding applications such as salty waters, fruit juices, and metabolic samples. Thank you very much for listening to this webcast, and if you have any further questions on these applications or any other isotopic water analyses, please feel free to contact us. You can reach Ian Green or Aaron Van Pelt at the numbers or email addresses shown.